Welcome to Pharmoon Drops. Multiple choice questions asked in different competitive exams are discussed here along with a brief description. First question of the episode is the analysis of biological fluids for sodium, potassium, calcium ions can be done using flame photometry. Flame photometry can be used for the analysis of biological fluids for determining sodium, potassium, and calcium ions. Flame photometry is also known as flame atomic emission spectroscopy. Flame photometry is also known as flame atomic emission spectroscopy. This branch of spectroscopy examines the species in the form of atoms. Flame photometry examines the species in the form of atoms. Flame photometry is used in inorganic chemical analysis. Flame photometry is used in inorganic chemical analysis. Generally, certain metal ions like sodium, potassium, calcium, and lithium are determined by using flame photometry. Generally, certain metal ions like sodium, potassium, calcium, and lithium are determined by using flame photometry. The procedure involves determination of intensity of the emitted light when the metal is introduced into a flame. The basic principle of flame photometry is the measurement of intensity of the light emitted when the metal is introduced into a flame. Wavelength of emitted light tells us what the element is, which means qualitative determination. Wavelength of emitted light tells us what the element is, that is qualitative determination. Flame photometry can be used for quantitative purpose also. Flame photometry can be used for quantitative purpose also. For this, the color intensity of the emitted light is considered. For qualitative determination, the color intensity of emitted light is considered. The emitted wavelength of sodium is 589 nanometer and the color will be yellow. The emitted wavelength of sodium is 589 nanometer and the color will be yellow. The emitted wavelength of calcium is 662 nanometer and the color will be orange. The emitted wavelength of calcium is 662 nanometer and the color will be orange. The emitted wavelength of potassium is 766 nanometer and the color will be violet. For potassium, wavelength emitted is 766 nanometer and the color will be violet. You can remember this by a word psychopov. I repeat, psychopov. C means SY. That means yellow color is for sodium. Co, that is CO. Here C is for calcium, O is for orange. POV, that is POV. Here PO is for potassium and V is for violet. Vitamin K is necessary for the formation of prothrombin. For the formation of prothrombin, vitamin K is essential. For the formation of certain coagulation factors, specifically factor 2 or prothrombin, factor 7, factor 9 and factor 10. Formation of these coagulation factors need a step, namely gamma carboxylation. For the formation of coagulation factors like factor 2, otherwise known as prothrombin, factor 7, factor 9 and factor 10, needs a biochemical step, namely gamma carboxylation. For gamma carboxylation, vitamin K is essential. For gamma carboxylation step, vitamin K is essential. Coumarin anticoagulants like warfarin inhibits gamma carboxylation step by inhibiting vitamin K action. Coumarin anticoagulants like warfarin antagonize the action of vitamin K, hence prevent gamma carboxylation step. The most commonly used vector in recombinant DNA technology is E. coli. E. coli is the most commonly used vector in recombinant DNA technology. Genetic engineering is otherwise known as genetic modification. Genetic engineering is otherwise known as genetic modification. It is the manipulation of organisms' genome using biotechnology. Genetic engineering is the manipulation of an organism's genome using biotechnology. It involves the transfer of genes within the species or across the species. It involves the transfer of genes within and across the species boundaries. Recombinant DNA technology, in short rDNA technology, is used to cut a known DNA sequence from one organism and introduce that into another organism. Recombinant DNA technology, otherwise rDNA technology, is used to cut a known DNA sequence from one organism and introduce it into another organism. rDNA technology can be used for producing monoclonal antibodies. rDNA technology can be used for producing monoclonal antibodies. Monoclonal antibodies are produced By cell culturing, monoclonal antibodies are produced by cell culturing. Generally, mouse spleen cells immunized with desired antigen are cultured. Generally, mouse spleen cells immunized with the desired antigen are cultured. And during that culturing, the fusing of myeloma cells with mouse spleen cells is done. During that culturing, fusing of myeloma cells with the mouse spleen cells is done. Polyploidy means the possession of 3 or more sets of chromosome. Polyploidy, that is Polyploidy. Polyploidy. Polyploidy means the possession of 3 or more sets of chromosomes. In other words, polyploidy is a condition in which the cells of an organism have more than one pair of chromosomes. In other words, polyploidy is a condition in which the cells of an organism have more than one pair of chromosomes. Polyploidy may occur due to abnormal cell division. Another reason for polyploidy is the fertilization of an egg by more than one sperm. Polyploidy may occur due to abnormal cell division or from the fertilization of an egg by more than one sperm. Histamine can contract bronchial smooth muscles. Histamine can contract bronchial smooth muscles. Histamine can stimulate sensory nerve endings in the skin. Histamine can stimulate sensory nerve endings in the skin. Histamine can stimulate gastric acid secretion. Histamine can stimulate gastric acid secretion. Commercial citric acid is produced by the microbial culture of Aspergillus niger. Commercial citric acid is produced by the microbial culture of Aspergillus niger. Citric acid is extensively used in the food and pharmaceutical industries. Citric acid is extensively used in food and pharmaceutical industries. Aspergillus niger or Candida species can be utilized for the synthesis of citric acid. Aspergillus niger or Candida species can be used for the synthesis of citric acid. Different sources of carbohydrates like molasses and starch-based media are used for this synthesis. Different sources of carbohydrates such as molasses and starch-based media are used for this synthesis. Fermentation of these sources with Aspergillus niger or Candida species produces citric acid. Carbohydrate sources can be fermented by using Aspergillus niger or Candida species to produce citric acid commercially.